So what we are doing here, we are basically taking out its DNA and we are selecting a segment of its DNA. Such segment or such particular DNA or nucleotide which will be not any harmful effect, not disease causing, not disease causing. Okay, so that DNA will amplify and we will insert that particular gene as an antigen into another harmless virus जो virus हमारे लिए harmful नहीं हो, that virus which will be not harmful for us. We shall import that particular segment to an harmful virus, sorry, harmless virus, which is not harmful for us, which is good for us. So here some recombination will happen, and when this virus will enter in our body, we shall give as a vaccine. Then its entire genomic material, it will be translated, and then new protein will be created, which will be like such proteins. So our immune system will be able to recognize it's a foreign substance. We have to remove this substance. So this is how you can produce the vaccine. Yesterday I have shown you a very interesting video. Today I do not have that much of time, but I will just show you plasmid. You can see जो हम लोग देख रहे हैं ना, ये बैक्टीरिया की बॉडी के अंदर की बात है, प्लाज्मिड. So by using enzyme, this plasmid or this particular, okay, in case of harmless bacteria, we can use bacteria's DNA. The plasmid is actually present in the bacteria. In case of your harmless virus, we shall use the harmless virus DNA. Okay, so we shall yeah, it was cutted, this part actually cutted, and then another gene. This gene is actually coming from the harmful virus, the virus which is harmful for us, like SARS-CoV-2. So we shall take that part of the genome, that part of the gene from the harmful virus, and we may insert it here. We may insert it here. You can see how, and then it will be fixed. And when it will be when this part will be, when this part will be synthesized, means transcription and translation will be happen. It will create protein, which protein will be actually similar to that virus, harmful virus, but not the such part of the harmful virus which is harmful. Okay, based on that, mRNA vaccines are actually nowadays come to the market. This is mRNA, and this is the proteins are actually formed. Based on that, various mRNA vaccines are actually come to the market. And yesterday, very interesting lecture, things I have discussed with your other friends. Those of actually have attended, they are so lucky. I told about the DNA fingerprinting. All of you are aware that nowadays, to detect the various crime, to solve various crime, scientists are using DNA fingerprinting. So how the things are actually used, I also told yesterday's lecture. So we are actually extracting some amount of DNA, then after from cell, then we are extracting the DNA, then we are actually that is this DNA will be this DNA pieces will be placed on the gel. Okay, for gel electrophoresis, this is the instrument. We shall place it here, particular this area. One will be negative, one will be positive charge, and based on that charge, when the machine will be activated, the DNA will be moved in this direction, and we can get such kind of various results. And there are based on that, there is something called STR. There is something called STR. Based on the STR, we will be able to identify. For that, we need the reference. If somebody is thief, इसने चोरी किया, उसका एक blood भी अगर बाहर पे हमें मिलते हैं, so we can that is a match that blood sample with the suspected people. अगर match हो जाते हैं suspected people की लोगों की जो particular the expression level के साथ, then he will be the guilty. Okay. I also told you in the video some example. Then on yesterday's lecture, I have told you about the making of insulin, how you can produce insulin in a large scale by using bacteria. ऋषभ, मैंने पूरा डिस्कस किया था ये चीज़, ये वीडियो दिखाया था या फिर ये पे खत्म कर दिया था?
वीडियो दिखाया था.
अच्छा अच्छा ठीक है. So I have told you on yesterday about how you can produce the insulin. So all of you are aware insulin actually used for the reduce the glucose level, है ना? Earlier time you can see earlier time peak pancreas are actually used for the production of the insulin. Insulin is actually already present in our body, which is actually coming from the pancreas islets of Langerhans, beta cell. But those people having some diabetes, they have a lack of they have a lack of insulin. They do not able to produce insulin, and glucose level is actually increased. So earlier time, pig pancreas are actually used for this particular insulin. Now after the advancement of genetic engineering, we are actually using bacteria. How? Very simple. We are taking the chromosome and we try to identify that which part of chromosome or which gene is actually responsible for making insulin protein. We take out that particular gene, insulin gene, and that gene will be actually inserted into the bacterial plasmid or DNA plasmid. And when that will be translated into bacteria, it will be give the insulin. So this is the picture of 1950 and this is a picture of now how scientists are using. पहले जो है काफ़ी सारे पीक्स को है ना मारा जाता था फॉर इंसुलिन. नाउ वी आर नॉट सेक्रीफाइंग एनी ऑफ द पीक्स. So I have discussed with a video on yesterday's lecture. I will not discuss again. And then very interesting thing called RT-PCR, PCR sorry. First thing is PCR. PCR से बाद में जो है वो RT-PCR आया. ठीक है, PCR is the first thing. So what is PCR? So I told you yesterday that this thing I will discuss on today because this topic is very interesting and all of you are supposed to know about the PCR, polymerase chain reaction. So you might have studied in your school days about the polymerase chain reaction, but in actual scenario, how the thing is actually working. Today we'll be able to learn. So let me first start with the basics. So you can see by using this is your DNA. Okay, this is your DNA. So you can make several copy of your DNA. For example, you have made one copy and another copy. From this copy, you can make two copies. And here this copy you can make two copies. From this copy, when it will be replicated, it will become 4. So similarly, you can get 16 copies from one DNA by using some enzymes and by using some methodology. You can get huge number of particular DNA segment. So using polymerase chain reaction, that is a minute amount of the DNA can be replicated very rapidly, producing enough DNA to easily detect and study and use in the variety of the application. PCR become a very essential tool in crime investigation, including diagnostic various genetic diseases. So why you are doing such kind of activities? We have one DNA. तो तुम लोगों के दिमाग में जो आ रहा होगा कि सर अगर DNA है, for example, you have mentioned here that crime investigation. So some crime actually is might have happened somewhere. So there are some dots, there are some drops of bloods. वहाँ पे जो है bloods की जो है कुछ drops मिले. So in that blood drops, there will be blood cells, and within that cell there will be nucleus, and within nucleus there will be chromosome and DNA. So it will be very easy to take that DNA and analyze its sequence and check this sequence is matching with which population. है ना? You might be thinking in such a way, but if you get a very small amount of particular sample, for example, like 0.1 millimeter, this is a very small amount. You know? In that small amount, you can imagine how much small amount of your DNA will be there. बहुत ही कम होगा, है ना? So that's why if your DNA sample is very small and if you try to get the sequence, it will be very difficult. It will be very difficult to analyze. And if you do any mistake, in that case, you will be not able to analyze the thing correctly. You will be not able to analyze the things in a correct manner. That's why we are actually producing several copies. We have some enzymes, we have some methodology. I will tell you more during my video presentation. I will show some video. So I will tell you more. So by using those enzyme and chemical, we can replicate that thing and we can get several copies. Now we shall analyze from that several copies. हम लोग जब इसमें काम करते हैं ना, हम लोग ऐसे नहीं करते कि जो है सिंपल हमारे पास वी गॉट वन डीएनए एंड फ्रॉम दैट वी विल जस्ट चेक. वी विल एक्चुअली बिकॉज देयर आर चांसेस ऑफ द मिस्टेक्स. वहाँ पे मिस्टेक्स होने के चांसेस जो है काफ़ी ज़्यादा रहते हैं. इसीलिए हम क्या करते हैं, हम काफ़ी कॉपीज़ जो है वो बनाते हैं. एक ही डीएनए का जो सेवरल कॉपी हम बनाते हैं, ताकि व्हेन वी विल टेक एवरेज और व्हेन वी शैल टेक that thing huge amount of that is samples, there will be less chance of mistakes. Okay, so ultimately this is the process by which we are actually producing our that thing, DNA sequencing. Now another thing I have to tell you, there is something called genome sequencing or DNA sequencing. So RT-PCR or PCR whatever you are actually using, this is actually coming later. RT-PCR ये बात की बात है, पहले ये चीज़ है. So basically after doing polymerase chain reaction, we are getting the sequence. We are getting the sequence P, C, whatever the things are, we are actually getting that thing. Okay. So then after that we actually compare our that thing with available already, our what are the already available sequence we have. We actually analyze with our already available sequences. Okay. So now come to very interesting part called RT-PCR. Yesterday I was actually telling that RT means real time. Somebody was actually saying, "Sir, RT तो हमने पढ़ा था कि reverse that is a transcriptase." And I was also not completely wrong. There was some electricity blockage. Am I audible to you, students?
Yes, sir.
Okay. There was a sudden electricity power cut was there. Okay. So let me tell you one by one, step by step, how RT-PCR actually being performed. So for example, this is the SARS-CoV-2. This is the SARS-CoV-2 virus. Let me remove the ink. So what we will do? First, the sample will be collected. Then after that, the RNA. SARS-CoV-2 is actually having the RNA, ribonucleic acid. It is not consisting the DNA. So we will extract that RNA. You can see this RNA is actually extracted here. And then reverse that is a transcription will be happen. That is from RNA to DNA will be make. We'll be making the RNA to DNA. So you can see there are two different that is a helix. Okay, from RNA to DNA actually being made here. Then making of the DNA template by using polymerase chain reaction. And after that, in third step, it will be a primer that will be bind to the DNA. Small primer sequence. Primer sequence are very important. It will be helpful for our understanding. It will be in my video. I will tell you more about that thing. Then DNA will be DNA is built of those primer. Okay, and after that, multiple copies of that DNA sequence will be created. You can see whatever I was telling you here. Same thing you can see, multiple copies of the DNA. Then after that, whatever the multiple copies you are getting, that multiple copies you are going to analyze by using fluorescence. By using fluorescence, you are going to analyze. And there will be some probe. One probe will be bind with other things. And through that, you will be getting one peak. You will be getting one peak. So if there is a lots of star that is a star or light, if it is at the top, it means you are actually COVID positive, because they are actually particularly checking that DNA. देखो यहाँ पे क्या चेक किया जा रहा है. मान लो somebody is actually not infected with COVID virus, and you are taking some sample from his that is a nose. You are taking some sample. But there is no any COVID virus. So in that scenario, there will be no peak. There will be no particular peak, because जो यहाँ पे जो डाला गया ना, whatever the things they have added, the probe and the primer, these are actually made for particular that virus RNA. ठीक है? Now there is no virus RNA. So in that case, there will be no any that is what I can say. There will be no any particular such kind of line. Okay, no probe found. It will be negative. So I have a video. Acha, another thing, we actually in last step we actually quantify the amount of light produced as a way of measurement of how much DNA is actually present. Okay, so that's why it is actually called also real time. It is also called real time RT. But actually, in terms of our enzymology, in terms of our enzyme, the RT at the second step, this step actually call it as a reverse transcriptase. Why? Because from DNA to RNA we're actually making by using transcription. Okay. Generally, in that is DNA to RNA is actually formed transcription. In reverse transcription, RNA to DNA is actually formed. यहाँ पे देखो ना तुम. In this case, there is a reverse transcription, RNA to DNA. So the process where RNA to DNA is actually formed. That's why it is called as a reverse transcriptase, a reverse transcription. So the enzyme actually responsible for making the RNA from DNA from RNA. That enzyme actually name is the reverse transcriptase, from where the RT name is actually coming. Okay, here in step 7 we are actually observing in real time that how much particular that DNA is actually that is present or its quantity. तुम लोगों ने देखे होंगे बहुत टाइम जो है ना, they actually tell the severity, how much severe the patient is or how much that is a COVID virus actually infected. कुछ ऐसा भी जो है डेटा वो लोग शो करते हैं. So this is possible because of the peak. The peak is very high than high number of your bacteria that is a COVID virus actually present. So by using fluorescence, they are actually giving you the results. So in real time or a quantitative measurements actually it is happening. That's why it is also called RT. So RT name जो है वो दो different जगह से आ रहा है. I hope it is very much clear to you all. Any questions from your side? I will show one video. I have a very interesting video about the RT-PCR. So कोई सवाल है तुम लोग पूछना चाहोगी before playing the video? समझ में आया तुम लोगों को? ऋषभ, पिंकी?
Yes, sir.
Okay. चलो, let me show you one of the video. Okay, let me make it zoom it so that you can. So after watching this video, you will be become expert. You'll be able to tell anybody that how the RT-PCR and other things are actually working. And this is the main goal of me as a teacher, to train you in such a manner so that you can become expert and you can tell people, the common people, those are actually not aware about the particular that virus and other things. Let me zoom properly. Just a second. Yeah, I hope it is very much clear right now than previous. Let me play. Rising here. I think system audio is not visible. Let me again share this thing. Yeah. Just a second, huh? Let me. Yeah.
Specimen. The swab is inserted in swabber and to start a test.
Can you hear the system audio also? The audio actually present in that video.
Samples
Yes,
can
sir. Yes, sir.
shortness of breath.
So all of you are aware about.
To start a test, the samples can be collected by a nasopharyngeal swab or an oropharyngeal swab. For nasopharyngeal specimen, the swab is inserted in the nostril and gently moved forward into the nasopharynx. Then it
So
is
this thing is also very important for you. There is something called nasopharyngeal and there is something called oropharyngeal. Naso में क्या होगा? Naso means nose, from here. From this area they'll collect, and oro means here. मुंह से जो है collect करेंगे.
rotated for a specified period time to collect
You can see.
secretions that contain the virus.
If it is virus possible present here, then
Once the swabbing is applied, the swab is placed immediately into sterile tube containing a viral transport medium. The standard method of coronavirus testing is polymerase chain reaction, PCR. which is a method that used widely in molecular biology to make millions to billions of copies of a specific DNA
You
fragment
can see.
rapidly. Coronaviruses contain genome. To detect these viruses with PCR, RNA molecules must be converted into their complementary DNA.
There is sudden electricity. Just a second.
The samples can be collected by a
अभी बार बार बिजली जा रहा है.
and gently moved forward into the nasopharynx. Then it is rotated for a specified period time to collect secretions that contain the virus. Once the swabbing is applied, the swab is placed immediately into sterile tube containing a viral transport medium.
See, in that sterile tube, all the particular compounds which will be making it stable. Okay, because of temperature, if virus is present, it might be get denatured. So such things will not happen.
The standard method of coronavirus testing is polymerase chain reaction, PCR, which is a method that used widely in molecular biology to make millions to billions of copies of a specific DNA fragment rapidly. Coronaviruses contain an extraordinarily long single-stranded RNA genome. To detect these viruses with PCR, RNA molecules must be converted into their complementary DNA sequences by reverse transcriptase.
Then see, RNA to cDNA, complementary DNA. I think somebody was asked one day one question, sir, what is complementary DNA? Complementary question है उसका opposite है ना, complementary DNA. So reverse transcriptase enzyme will be used.
Newly synthesized DNA can be amplified by standard PCR procedures. This approach is universally known as RT-PCR. To perform this method, basically viral RNA should be extracted. Several RNA purification kits are available for convenient, fast and effective isolation.
You can see.
To extract the viral RNA by using
collected now from that nasopharynx or oropharynx. That sample was actually kept here. Then from this particular that is a pipette, this is a micropipette. ये जो है ना इसको micropipette कहते हैं इसको. So by using that micropipette, the samples are actually collected and it is actually placed here. Okay, इसमें हाँ, yeah.
Commercial kit. The sample is first added into a microcentrifuge tube. Then it is mixed with a lysis buffer. This buffer is highly denaturing and usually consists of phenol and guanidine isothiocyanate. Also, RNase inhibitors are usually present in the lysis buffer to ensure isolation of intact viral RNA. Once the lysis buffer is added, the tube is mixed by pulse vortexing and incubated at room temperature.
Separate the other things.
Then the virus is lysed under the highly denaturing conditions provided by the lysis buffer. Once the sample is lysed, a purification procedure is carried out by using a...
You can see that picture, the main reason centrifugation actually done just to separate all those things so that RNA can be extracted very easily. Okay?
The sample is lysed. A purification procedure is carried out by using a spin column. The sample is loaded onto the spin column. Then a centrifugation is performed. This procedure is a solid phase extraction method, in which the stationary phase consists of a silica matrix. Under optimal salt and pH conditions, RNA molecules are bind to the silica gel membrane, and at the same time,
RNA molecules are present, then other various like your spike protein, AMPRO, 3CL Pro, whatever the proteins are present in that virus, SARS-CoV-2, all the things are present. So only your RNA, that thing will be bind with the silica.
Protein and other contaminants are not retained.
Sirf RNA or silica.
After centrifugation, the spin column is placed into a clean collection tube, and the filtrate is discarded. Then a wash buffer is added. The column is put in a centrifuge again, forcing the wash buffer through the membrane. This removes any remaining impurities from the membrane, leaving only the RNA bound to the...
See, any impurities may present. For example, other proteins, spike protein, etc. If any protein is present, it will create a problem. So that's why, in this case, it is they are doing for second time.
Silica gel. Once the sample is washed, the column is placed in a clean microcentrifuge tube, and an elution buffer is added. Then, a centrifugation is carried out, forcing the elution buffer through the membrane. The elution buffer removes the viral RNA from the spin column.
Now comes
And a
to
purified RNA, which is free of protein, inhibitors, and other contaminants is obtained. After the extraction of the viral RNA, the next step is
you
the
might be wondering that sir why they have done the centrifugation for thrice. So पहले दो बार जो किया just to get the RNA from the mixture of the that is other cell nuclei, other cell components. In third steps, they have done the cleaning just to remove the RNA which is actually adhere to the silica.
preparation of the reaction mixture for PCR amplification. In this step, a master mix is used, which is a pre-mixed concentrated solution that consists of buffer, reverse transcriptase enzyme, nucleotides, forward primer, reverse primer, Tachman probe, and DNA polymerase.
Now you might be wondering, sir, what are these things? Just a second. What are these
DNA
various things?
polymerase.
The RT, reverse transcriptase, D, NTPs, reverse primer, forward primer, Tachman probe, and DNA polymerase. See, you have no need to edit. Right now, after doing all such things, you have the RNA. So you have to convert that RNA into DNA. For that, you need some enzyme. So the enzyme, what the enzyme will do? The enzyme will make the RNA based on these primers and other things. Okay. So in that case, the DNA polymerase, reverse transcriptase, then your that is a D, NTPs, all these things will be come to the picture. So you added
Finally,
master
to
mixer.
complete this reaction mixture, the RNA template is added.
You add the RNA,
The tube is mixed by pulse vortexing.
and in both.
Then the reaction mixture is loaded into a PCR plate, which generally contains 96 wells, allowing the analysis of several samples at the same time.
So can you imagine how difficult the
Next,
test is?
the plate is placed in a PCR machine,
This
which
the
is
PCR
essentially a thermal cycler. Real-time RT-PCR is used for the detection of the new coronavirus 2019 by the amplification of target
So
sequence.
here lots of things will be happen. Here lots of things will be happen. First it will be converted into the DNA, and then it will be one by one. It will be that is copied for millions and billions time, and then it will be analyzed by using the fluorescence, whatever happens.
In the RDRP gene, the E gene, and
This is
the
the
N
entire
gene.
genome.
The choice of the target gene depends on the primers and the probe sequences. The first step in RT-PCR is reverse transcription. The first strand complementary DNA synthesis is primed with the PCR reverse primer, which hybridizes to a complementary part of the virus RNA genome. Reverse transcriptase then adds DNA nucleotides
see
onto
one
the
by
3'
one, this is
end of the
added.
primer, synthesizing
that you
DNA
can see.
complementary of the viral RNA.
In our previous video we have seen that there are lots of probes, etc., etc. So you can see these probes are actually falling here, and your DNA is actually forming.
The temperature and duration of this step depend on the primer, the target RNA, and the reverse transcriptase used. Next,
Complementary
an
DNA.
initial
Now, this is
denaturation
again.
step is applied, causing denaturation of the RNA-DNA hybrids. This step is required for the activation of DNA polymerase and simultaneously the inactivation of reverse transcriptase. PCR consists of a series of thermal cycles, with each cycle consisting of denaturation, annealing, and extension steps. Denaturation step consists
The
of
first
heating
step,
the reaction chamber to 95 degrees Celsius, and it is used for denaturation of the double-stranded DNA template. In the next
Then
step,
annealing.
the reaction temperature is lowered to 58 degrees Celsius, allowing annealing of the forward primer to its complementary part of the single-stranded DNA template. The annealing temperature relies.
See, this part actually coming from our that viral RNA, है ना? DNA was formed. Now that part actually is actually synthetic. वो हमने बनाया है. वो वायरस से नहीं है, वायरस का नहीं है पार्ट. हम लोगों ने जो है वायरस के जो RNA है, from that we actually got this. Now these things will be help to make multiple copies.
Relies directly on length and composition of the primers. In the extension step, the DNA polymerase synthesizes a new DNA strand
You can see.
complementary to the DNA template strand by adding free nucleotides from the reaction mixture that are complementary to the template in the 5' to 3' direction.
I hope now it will be very much clear to you that how we actually copy from one DNA to multiple, that is one RNA to multiple RNA copies. Okay, these things will again use as a template.
The temperature at this step depends on the DNA polymerase used. After the first cycle, the double-stranded DNA target is obtained. Then, the denaturation of this double-stranded DNA is performed, yielding two single-stranded DNA.
Cycle 2, again we actually that is a breaking, and then this part will be again used as a template, and it will be again form as a another copy.
DNA molecules. In the next step, the reaction
So DNA
temperature is
selection,
lowered,
annealing, and
allowing
extension.
annealing of
You
the
know, continuous
primers to
thing
each of the
will be
single-stranded
one
DNA
and one and
templates,
on,
and annealing
and
of
when
the
the copies
Tachman probe to its complementary part of the target DNA. Tachman probe consists of a fluorophore covalently attached to the 5' end of the oligonucleotide probe. The fluorescence is emitted by the fluorophore when it is excited by the cycler's light source. Also, this probe consists of a quencher at the 3' end. The close proximity of the reporter to the quencher prevents detection of its fluorescence.
So I
In the
told you
extensions,
that we also have to analyze whatever the multiple copies are actually forming. We have to analyze that copies under fluorescence. So for that is a analyze under fluorescence, we need Tachman probe. So Tachman probe is actually this R and Q. So whatever the from R, if R is present, from here your fluorescence will be activated and it will be analyzed. Okay, you will see.
step, DNA polymerase synthesizes new strands. When the polymerase reaches a Tachman probe, its endogenous 5' nuclease activity cleaves the probe, separating the dye from the quencher. With each cycle of PCR, more dye molecules are released, resulting in an increase in fluorescence intensity
You can see.
proportional to the
Now it will
amount
be
of
very clear to
amplicon
you.
synthesized. This method allows the estimation of the amount of a given sequence present in a sample. The number of double-stranded DNA pieces is doubled in each cycle, therefore, PCR can be used to
So you have
analyze.
to equal is another thing. This number of cycles you can increase. Okay, number of cycle you can increase. You might have heard that some minister, even a minister or a very senior person got infected or showing some symptom of COVID-19. In that scenario, they are taking much more time for RT-PCR. Basically, they are taking much more time means they are using multiple cycles. Okay.
Is extremely small amounts of sample.
So that huge number of copies can be created. You can see copies of a target DNA कितना create हुआ 2 घंटा. They have taken 2 hours. So RT-PCR is actually time consuming process. It is not antibody or antigen test where you will be getting the result as soon as possible. And
For the
then
measurement of the fluorescence
is the
signal,
last step.
a tungsten halogen lamp, an excitation filter, mirrors, lens, an emission filter, and a charge-coupled device CCD camera are used. Filtered light from the lamp is
This is
reflected
the
off
arrangement
mirror,
you have to
passes
do
through a condensing lens, and is focused into the center of each well. Then fluorescent light emitted from the wells reflects off the mirror, passes through an emission filter, and is detected by the CCD camera. In each PCR cycle, light from excited fluorophore can be detected by the CCD camera, which converts the light that it captures into digital.
So whatever the CCD camera actually captured, that thing will be actually coming in your the computer screen. Here you'll be getting some curve that
Digital
will
data.
show you.
This method is known as real-time PCR, which allows the monitoring of the progress of the PCR reaction as it occurs in real time.
You can see. So I hope it is very much clear to you all. और
PCR reaction as it occurs in
जाने होंगे कि how the things are actually happening. Okay, so whatever I was actually discussing you here, so that thing you have seen in the video in a very perfect manner. So I hope you can able to write, you can able to explain to other people also that how RT-PCR actually working. So in future, if you want to make your own RT-PCR type of test for some other diseases, you can also make. For that, you just need this thing, this thing, the reverse transcriptase or particular DNA and primer. Okay, so that's it from my side on today. Baki, I have another example that is not so much important for biopharmaceutics, for pharmaceutical point of view. This is about the how you can grow the plant by using genetic engineering at the dark room. China last year China actually sent their some mission in moon मून MWN मून में जो है मून में जो है चाइना ने अपना एक मिशन भेजा था कहाँ पे मून की इन दैट मून देयर इज सम एरिया where sun that is rays are actually not reaching. This is the dark area in the moon. Last year China sent one moon mission at the dark side. They have tried to see whether plant can be grow in the dark side or not by using the same technology. Let me show. The scientists those are actually growing the plant. You can see this is a plant.
a novel plant that glows in the dark using the latest tools and techniques that are known as synthetic biology.
So in the dark place, the plant is actually growing. You might be wondering, that's a photosynthesis कैसे होगा? इसका भी solution है ना? Without light.
I must admit, the moment we could actually see it and get it over the visibility threshold was one, a huge relief, and two, like, oh, thank God.
There's some accreditation, there's some things.
Really, the goal of that glowing plant project was to create a product that symbolizes the advances that we're making and inspires people to imagine what else we can create.
The conversation should be like, how should we use this tool, as opposed to the knee-jerk reaction of.
So this is the plant. So let me tell you that how actually they have done. They actually make some changes in the genetic level. They have made some changes in the genetic level of that particular plant. But yes, this is not very useful. ऐसा नहीं हम हर एक plant पे जो है process use कर सकते हैं. No, it's not gonna work for every plant. So there are some plants which are actually growing at the dark places. So they have actually used that gene, and they actually fused that gene by using recombinant DNA technology with those plants which plants are actually growing in the light. Okay, due to that the plants which are growing in the light, they are also growing in the dark. Okay, this is the simple phenomena.